Biomolecules. How to analyze chemical composition, primary and secondary metabolites, biomacromolecules are proteins, polysaccharides, nucleic acids, structure of proteins, then enzymes. So how to analyze chemical composition? A comparison of elements present in non-living and living matter. So element hydrogen, carbon, oxygen, nitrogen, sulfur more in human body than earth crust. This is percentage weight of earth crust versus human body. And sodium, calcium, magnesium, silicon more in earth crust than human body, although nitrogen negligible in earth crust and silicon negligible in human body. So this is a comparison of elements present in non-living and living matter. A list of representative inorganic constituents of living tissues components: sodium, potassium, calcium, magnesium, water compounds, NaCl, calcium carbonate, phosphate, sulfate. Now alpha amino acids. Now there is deuteronic form, contains NH and H3 plus and COO negative, NH3 plus. So now coming to this, like diagrammatic representation of small molecular weight organic compounds in living tissues includes glucose and ribose. So CC Castrol O6 glucose, see the diagram, OH at 3 degree on top, and in ribose all the OH on bottom. So C5H10 O5, this is ribose, these are the sugars, these are carbohydrates. And then amino acids, that is glycine when hydrogen present, CH3 when alanine, serine when methyl, when hydroxymethyl, CH2OH hydroxymethyl present. So these are amino acids, 20 amino acids. Then fatty acids is type of lipid. Fatty acid, palmitic acid, COOH, CH2 14 and CH3. Now glycerol, kind of alcohol, hydroxypropane, CH2 CH, CH2 OH, OH, OH. So triglyceride, 3 fatty acids joined to glycerol. So these are triglyceride, this type of lipid. Okay. And then phospholipid, lecithin, found in cell membrane. You see two fatty acids and then phosphate group and then choline. So this is phospholipid, that is lecithin, is also a lipid, type of phospholipid. And then cholesterol, kind of steroid, which is also a lipid. So OH and 4 ring present. Cholesterol is a homocyclic. So fats and oils, all these are lipids. Okay, now coming to this, in DNA there are polymer of nuclearotides, so contains nitrogen bases, can be purine and pyrimidine. In adenine you see 2 heterocyclic ring, and then in uracil 1 heterocyclic ring containing 2 hydrogen 2 nitrogen atom. Now in nucleosides you see, so in nucleosides you see adenosine and then uridine. So all that uracil, adenine, OH on the bottom, and then CH2OH on the top. So adenine and uracil, so these are nucleosides. Okay, and then nucleotide, you see adenylic acid, adenine, OH and phosphate and OCH2, OH and O. So these are the nucleotide. So diagrammatic representation of small molecular weight organic compounds in living tissues. Now primary and secondary metabolites. So primary and secondary metabolites you see. So primary metabolites involves in primary function. So secondary metabolites. So secondary metabolites includes alkaloids, flavonoids, rubber, essential oils, antibiotics, color, pigments, scents, gums, spices. Now some secondary metabolites: pigments, carotenoids, anthocyanins, alkaloids, morphine, codeine, terpenoids, monoterpene, diterpene, essential oil, lemongrass oil, toxins, urbane, resin, lectins, concannabidiol, air drugs, vinblastine, curcumin, polymeric substances, rubber, gums and cellulose. And then biomacromolecules differ from biomicromolecules Average composition of cells component percentage of the total cellular mass, water 7 to 19, and then proteins 10 to 15, carbohydrates 3, lipids 2, nucleic acid 5 to 7, ions 1%. And in protein, some proteins and their functions: protein collagen functions, intercellular ground substance, trypsin, enzyme, insulin, hormone, antibody, fight infectious agents, receptor, sensory receptions for smell, taste, hormone, etc. Glut enables glucose transport into the cells. Now in polysaccharides there is glycogen. It is a branch polysaccharide. It is type of carbohydrates which stores the glucose. Branching is 1 alpha 1,4 glycosidic linkage and 1,6 alpha glycosidic linkage. You see in the diagram. So nucleic acid, then structure of proteins.